Maria. Hello, dear respected colleagues, dear members of the network and other participants in this webinar. This is the 19th webinar that is organized inside the INSAS network. And in this case, the presenter will tell us everything about a model called the SALTMED model, that is an integrated management tool for water, crops, soil salinity, and nitrogen fertilizers. Allow me to introduce to the speaker. It is my pleasure. It is an old colleague. The speaker is Professor Dr. Raghav, that is Honorary President of the International Commission on Irrigation and Drainage, and Fellow Principal Hydrologist and Water Resource Management Specialist at UK Centre for Ecology and Hydrology, Wellington, Oxfordshire, United Kingdom. He is now honorary president, but he was president from 2020 to 23 of the International Commission of Irrigation and Drainage, vice president in the period 2010 to 2013 of the International Commission on Irrigation and Drainage, chairman of the Permanent Committee on Strategy and Organization, chairman of the Working Group on Water, Food, Energy Nexus, ICID, still this chairman of this working group, and chairman of the British National Committee on Irrigation and Drainage. He's founder of the working group of the use of non-conventional water resources for irrigation, adjunct professor at Alexandria University, Egypt, from 27 to present. He acts as well as international advisor to several national and international organizations, lecturer, supervisor, and external examiner to various universities in and outside the UK, evaluator for several funding organizations, and member of promotion panels of several universities. Professor Raghav is recipient of several national and international awards, reviewer for more than 20 international journals, author and co-author of numerous scientific papers, an editor and co-editor of several special issues of international journals, as well as conference proceedings. Member of the editorial boards of a number of scientific journals, including the Cambridge University Journal of Agricultural Science. Professor Dr. Raghav developed two widely used models, SALTMED, that is the topic of this webinar, for field scale, as a tool for integrated management of water, soils, crops, and nitrogen fertilizers. And another model, the IHMS, Integrated Hydrological Modeling System, for catchment basin scale. He tells us that both models are freely available to download from the International Commission of Irrigation and Drainage website. His areas of work include hydrology, irrigation and drainage, remote sensing applications, urban hydrology, climate change impact on water resources, uncertainty analysis of predicted river flows, modeling at field and catchment scales, human intervention impact on water resources, management of non-conventional water resources, water food energy nexus, crop irrigation water requirement determination, water saving technologies, and water harvesting at different scales. He is author of an impressive number of publications. And it is especially appreciated that Professor Dr. Raghav will deliver the webinar even suffering of flu. And he apologized if some some symptoms of this are evident during the webinar. Thank you very much, Professor Dr. Raghav, and the floor is yours.
Thank you, Professor George, the president of the International Network of Salt-Affected Soils. It's a pleasure for me to give this webinar today. I will start by talking about the main processes and equations of the SALTMED Then I'll try to go through the model frames and explain them. And if we have enough time, I will do a demo with the model and look at some worldwide applications. See how it goes with the time. Thank you. I will start now by sharing my screen. So the first part now is the processes and the equations. This is really the opening slide of the SALTMED. As Professor George mentioned, it's a free download from the ICRD website and also from UKCH website.
Excuse me, sir. Your screen is not visible.
Okay. Let me do the sharing again.
Don't worry, the number of participants is increasing. So it's a time of profit for the webinar.
Yeah.
It is now in working mode. Perfect.
Okay.
Thank you.
All right, thank you. So I'll start by talking about the equations and the processes that really we have in SALTMED. This is, of course, the webinar organizers' acknowledgement to especially the International Network of Salt-Affected Soils who invited me to deliver this webinar. The processes and equations I will start with by looking at, this is really what you will see when you run the SALTMED as really something you will see, and the download is here from the ICRD as well as from UKCH. There is development of that model started by 2002, where the first paper really appeared about the model, and it was the outcome of EU-funded project SALTMED, and that project where the name of the model came from was coordinated by Professor Tim Flowers from Sussex University, UK. And then in 2015, more development appeared and it was published in 2015. And then there were some other more detailed descriptions published in 2023 in ebook, as you see on the list here. But also there are two special issues published in relation to the model in 2005 and also in 2010 Saphir project. And in 2020, Wiley published a virtual special issue, combined about 17 research papers on SALTMED. And these are really the special issues that focused on the applications of SALTMED. And also we, as part of the Water for Crops project and having as a partner a consultancy company from Germany, Aachen, Germany, and also they are associated with University of Aachen, they produced online course for people that they can really do the course online. And it has part how to install the model, the whole course about different parts, really, how to run, how to get the data, and also troubleshooting if you have a problem and so on. And all these are put together in YouTube, several videos. The rationale behind the development of SALTMED was really to study the impact of using saline water on field crops of the Mediterranean region and to assess the short and long-term impact of using saline water on the environment. And then the model has been developed forward after that with other projects came after SALTMED, like Saphir, like SwapMed, and Water for Crops. The model has been successfully tested against experiments in several countries. You can see them on the list here. And it has also been used here in the UK to look at the impact of seawater rise and inundation of some 7 lowland coastal sites. The model actually is suitable for academic research and for professionals and for practitioners. So each one of those will find something in that model of interest. The benefit of using SALTMED model is to help really predict the impact of climate change, rainfall temperature, CO2, seawater intrusion, seawater inundation, tsunami, on soil vegetation and food security, and also to improve the water use efficiency, reduce water use in agriculture, and increase productivity. It helps also to select the best strategy to irrigate using less water and save more. For example, using the PRD and can save up to 40% of water. Also can guide user to select the most suitable crops and irrigation system and irrigation strategies when especially using saline, brackish water, or even treated wastewater. And also to predict the poor quality water on environment and to guide the user to the best strategy to minimize the negative impact of using poor quality water. The model has different components, and these are the components of SALTMED: the evaporation, calculations using different methods, plant water uptake, accounting for salinity and water stress, water and solute transport under different irrigation system, soil nitrogen cycle, including soil temperature dynamics, leaching of salinity and nitrogen fertilizers, drainage flow and the groundwater level dynamics, crop yield, biomass, water use efficiency and productivity, crop rotations of different crops, simultaneous runs of up to 20 multiple fields or treatments. The model has crop and soil databases and example files, and the model can run with what-if climate change scenarios, and the model calculates the crop growth based on degree days and calendar dates, and the water balance, salt balance, and nitrogen balance are provided also by the model on a daily basis during the run. And when you look really at how many fields or how many different treatments, and this is really a part of the model importance is that you can run up to 20 different fields with different irrigation systems, with different crops, or even different nitrogen treatments levels or different salinity levels, up to 20. But of course, that depends on the speed. Will depend on how good your computer memory and processor is. So the equations now were used in the SALTMED model for evaporation, for example. The calculations options are: we calculate the reference evapotranspiration ET node from class A pan measurements. If you have only class A pan, so you could still use SALTMED. You can also calculate it ET node from the modified Penman-Monteith equation, modified by FAO 1998. You can also use the original Penman-Monteith equation, but you need, of course, to put the canopy resistance as input. And there are options also in the model how to get the canopy resistance as input. You can calculate the canopy resistance from equations based on environmental parameters, and I will show that in a minute. Or you can use the abscisic acid and as a plant hormone concentration and leaf water potential measurements to calculate the canopy conductance. You can also get the canopy resistance from field measurements, or you can simply give a seasonal average value. So there are different ways. If you want to use the original Penman-Monteith equation and you are stuck with the canopy conductance, there are several ways to get around it. So I will start with the evaporation. This is the class A pan, and all of us used it in the past. The other option, of course, is to use the original Penman-Monteith equation here. And you can see here that that's actually one. Most of these parameters are weather parameters related to the weather, but the RS here is a factor that really represents the plants and biological control on the transpiration and evapotranspiration calculations. So I will not go into these details, but there are some people try to calculate the canopy resistance or conductance from environmental factors like this equation of Jarvis and Corner, where they calculate the conductance here from different factors like vapor pressure deficit, temperature, soil water, and radiation. There are other ways that they can calculate the canopy conductance using the hormone the plants would release, especially under stress, that the ABA or abscisic acid that goes from the roots to the stem and to the leaves, and asking the leaves to close partially the opening so not to allow losses by transpiration while accepting enough CO2 for biomass accumulation. So there are some scientists, especially I came across in Lisbon University, and they were interested in this sort of academic type of research, is to get the canopy conductance from the biochemical signal of the plant that controls the losses by evaporation from the leaves. So you could do that, and there is equation here and developed by Tardieu et al., and you can calculate the canopy conductance or resistance from the abscisic acid and the leaf water potential. And the FAO, in collaboration with ICID, they found another way to get around the canopy conductance by simply, instead of the usual canopy conductance that goes up and down during the day and the night and also over the season, to a fixed value, 70 seconds per meter. And by this, they modified the original Penman-Monteith to the modified Penman-Monteith that assumes the resistance, canopy resistance is 70 seconds per meter and an average seasonal value. And that led to this equation. So most of the people, they're using this equation, and you get the reference evapotranspiration. And if it's multiplied by the crop coefficient, which is divided into two parts, one represents the soil evaporation, one represents the transpiration of the plant, and then you can get the crop evapotranspiration. Another process in SALTMED is the water uptake, especially in presence of salt. And the equation used in SALTMED is the equation suggested after so many years of development before that by Cardon and Letty. And here the actual uptake is related to the maximum uptake under optimum conditions, stress-free. And it takes into account the presence of salt here represented by pi, and also water stress represented by H here. And in the model, we also realized there is a stronger relationship between the uptake and the yield. And we try to represent that in a form of relative yield, which is the relation between the total seasonal actual uptake divided by the potential uptake. And this is relative yield. And if some people would like to have an idea what will be the actual yield, you can multiply relative yield by maximum yield observed in the area under optimum condition. That option, actually, we use it only for scenarios. There are some people recently, I saw them uh, using that option instead of the main way of calculating the biomass. So this is, I will insist here to mention, this is only for scenarios, and there is another option to calculate the yield properly from the biomass and the photosynthesis. That will be the next. So that's the way we calculate the yield and the biomass, and basically it is related to the assimilation, and the net assimilation is assimilation by the photosynthesis minus respiration losses by the plant due to the increase in temperature. So the assimilation rate per unit area is basically photosynthesis efficiency multiplied by the radiation and then affected by other factors. They might reduce actually the biomass production like temperature and transpiration and the amount of nitrogen in the leaves, as the leaves actually is the factory where the biomass starts. So basically it is really photosynthesis efficiency, radiation, and stress factors related to temperature, transpiration, and the leaf nitrogen. And that's how really we calculate the biomass and the yield. And these actually the factors that affect the biomass, that plants usually they grow at optimum temperature when the temperature between that level and that level. And then, of course, the growth will go to zero at maximum temperature, and there is a minimum temperature also for the growth. And when it comes to the water stress, also the plants, they don't really grow at wilting point, and they grow happily when the water goes more towards field capacity, and they can stay here for longer time, and that depends on the type of plants, really. One can have that wide plateau, another plant will have that narrow plateau, but yeah, the stress actually can go also down to saturation. When the water is saturated, of course, it would affect the growth, as the oxygen is also needed for the growth. What the model actually SALTMED deals with in terms of water and solid movement, it deals with several irrigation systems. For example, here, this is one-dimensional water and solid flow, and this is what you will see when the model runs. And that's like a basin irrigation, flood irrigation, and this is one-dimensional flow. When you have a trickle line source of drippers or furrow irrigation system, this is what you'll be able to see when the model runs. And if you have only one dripper um, uh, beside the tree or far away from the other dripper, then in this case you will have the 3D or cylindrical flow where the water and solute are described. So in rain-fed agriculture, sprinkler, irrigation, water, and basin, these are one-dimensional water and solid flow model. Furrow, trickle, line source, water source transport can be described by two-dimensional plane flow and Cartesian coordinates X and Z. The trickle point source, water and solute, can be described as three-dimensional and can be viewed as cylindrical flow, and coordinates would be R and Z instead of X and Z. And this is an example of how we calculate the flows. And if you have here a trickle point source at that point, we divide the domain into squares. For the moment, the default is 4 by 4 centimeter. And we calculate the change of the soil moisture or solute concentration on the middle compartment here from what goes in and what goes out from the four adjacent squares. And when it comes to water flows, we follow the Darcy law, um, where the water flows according to hydrological gradient from one end to the other, multiplied by the conductivity. And the change in the soil moisture content within 1 unit of the soil is based on the input minus the output, and also minus the water taken by the roots. And that will give us the changes in soil moisture content based on the continuity equation. So that's a type of Richard equation that calculates the change in soil moisture in one direction in presence of the plant, where S is the water uptake. And when it comes to really solute, they have um, diffusion and they have a dispersion. So when the solute flow by convection, it means that the solute moves with the water, then it depends very much on the velocity. So it's the flow and the concentration. And when the water is at rest, then it follows Fick's law, which means that you have a concentration gradient, uh, but with water not moving. So this actually the convection flow of solutes and the diffusion flow of solutes, they can be combined together. And that there is something happening there. It's called dispersion. When you have a molecule moving between particles, in the middle here, you have no friction between these two. So the velocity here is quite high. And here, because of the friction between the molecule here and the soil particles, that actually reduces the speed. And that sort of a speed or velocity distribution creates a difference in concentration due to the water and solute movement in that direction. And that's what we call a dispersion or mechanical dispersion. And also, when you have a soil moisture, the soil moisture here level can affect also that dispersion and the differences in concentration due to the movement. So we need to combine the two flows by having really the convection, and also we having the dispersion and the diffusion. So basically, the overall solute flow is diffusion plus dispersion plus convection flux of solute. And that's what's considered in the model. And also we take into account that the solute diffusion in the soil affected by the soil moisture. And the soil moisture also has something to do with tortuosity, because the film around the particles that makes the molecule might go a longer distance when you have less soil moisture. So taking these into account, so for one-dimensional solute flow, we can see that sort of convection dispersion equation that everyone knows, where it has got here hydrodynamic dispersion coefficient. So this is really a combined diffusion and a dispersion coefficient in one parameter. And this is the convection flow that solute moves with the water, but this actually the solute moves according to the gradient or the difference in concentration, and also the uptake of the solutes. It could be nitrate, could be ammonia, by the plants is also taken into account. And for the transport of 2D, now we have to consider the movement of X direction and also in Z direction. And that's the equation for the water when the water moves into furrow irrigation or trickle line source, or the drippers, they overlap the wetting fronts over a short period of time. But when it comes to the solute movement, it's the same convection dispersion equation, but here it becomes really more of an issue, because the dispersion and diffusion equations combined here, they are really need to be obtained, and some people they measure them, and some of them they obtain them by a different means or literature or by calibration. So in the model, you can, from these equations, you can get the soil moisture content, you can get the salt concentration, you can also get the salt content, you can also get the relative salt concentration. Soil hydraulic properties in SALTMED is based on Van Gnuchten equation. And this is the equation that we use based on Van Gnuchten. And these are input values. Either you put them as a user or it comes from the database of the model. And this is for the relation between soil moisture and what metric potential, or the PF curve, if you know. And this is the unsaturated hydraulic conductivity, and as a function of also the matrix potential. And there are also options in the model that you can put your own data, like a PF values, soil moisture against metric potential, hydraulic conductivity against metric potential. You can also use that if you measure them in the lab or in the field. The salinity leaching requirement is calculated in the model as difference between the concentration of irrigation water to drainage water. And in case if you don't have drainage water, you can take the concentration of the root zone as a mean value. And also in the model, we have subsurface drainage, open and pipe drains based on Hooghouten equation. And also in the model, you can have these open and closed system based on this equation. And in the model also, you can change the hydraulic radius and wetted permeameter according to the shape of your drain system. Additional thing that's also added to the model after the original one of 2002 is the nitrogen cycle. A nitrogen cycle based on the soil N model of Uppsala State University of Sweden, where I had the chance to be working with that group for 1 year. So the soil N model looks at all the transformation that happens here and for the nitrogen cycle in the field. And these are mineralization, immobilization, nitrification, denitrification, leaching, and plant uptake. And most of these actually mechanisms or actions, like the mineralization of humus, are all of them first order rate equations. So basically it is amount multiplied by factors related to impact of moisture or impact of temperature, plus a rate constant, which is the rate at which the humus, for example, would be mineralized to ammonia. So this is, for example, of how the mineralization of humus to become ammonia looks like. And here you use humus amount multiplied by rate constant and impact of temperature and soil moisture. And the same operations, the same way for others. So here the general equation is the amount, mineralization rate, and multiplied by the effect temperature and effect of soil moisture. The same thing, mineralization of litter, and the litter can be formed, can be mineralized to CO2 or humus or litters or ammonia as well. So there are different ways really for mineralization of litter. And nitrification, where the ammonia would be transferred to NO3, is the same first order rate reaction. It's amount multiplied really by factors that affect the mineralization or nitrification rate. And here denitrification of NO3 to N2 and N2, and also the same equations. Then we have also the plant nitrogen uptake, and this is based on Greenwood equation that gives you the amount of nitrogen taken by the plant, whether it's in form of NO3 or NH4. And the model, when you install the model, the model will show you these subfolders under C and SALTMED data _ data. That's what you should see, actually, all that. You might not see those. I created myself during the runs. But you should have here all these folders when you install. Try to install the model on C rather than other drives, because your database is actually saved in most of the computers under the C program files as Access Microsoft. The model input is in different formats. It could be Excel files, it could be Access database, it could be text files. Parameters, values, like Pi 50, for example, new soils, new crops. You can put these parameters, you type them in and save them either as a text file or into the database. The results also can be given as Excel, separated variable files, or as an Access database if requested, or text file. There are some links to the model. As I mentioned early on, is the one link that you download the model from ICID or UKCH, and also the online course, which I advise everyone to listen or to watch the online course. We did organize physically that course to groups from all over the world in UKCH, and people, they came, and it was 3 days course. So what I'm really presenting today is something that we give over 3 days course, physical presence. And these are also some people, when they run the model, they get problems. And you need to look at these actually different YouTube videos, one for each problems that we expected the user might see. This is real-time screen graphic output you will see when you run the model. For example, you will see that on the trickle line source, soil moisture, you can see soil salinity. You can see also those profiles of nitrogen, horizontal and vertical. Under subsurface drip irrigation, you might also see that plots for every day model run. This is under a basin irrigation system. You can also see the soil moisture profiles and salinity profiles. If you have a different layers of the model, so it means your soil is layered, not homogeneous, you can still see the impact, how it would look like if your soil is not really homogeneous all the way down. And you can see that also in the soil moisture reflecting, you have really 3 different layers. When it comes to PRD, or partial root drying, where we irrigate, and instead of irrigating the plant every time with the 2 drippers, the plant here is in the middle, and drip line on the right, drip line on the left. And instead of getting these 2 drip lines to work all the time, we irrigate one time was on the right side and another time was the left side. So we alternate. So basically, we irrigate half of the root zone. So that's how it should look like in the field, and that's actually part of SALTMED now in the latest version, not in the original one. And this is what you should be able to see. And here in the plot, you can see that dripper here is the one in action now, which is receiving water, while the drippers away on the other side here is not really in operation right now. So you can see that. So now it's the other way around. So the dripper here stopped irrigating because now it's the turn of the other dripper to start working and operating. And when you look at the soil moisture profile, you can see something like a seesaw when the dripper on the left works. So that actually a line will go up and that line would go down. and the other way around. So you can see from the plot of soil moisture which dripper is working. The KC factor, we get the values from the user, whether it's based on FAO 56 or user input, and it comes as a stage value, and we interpolate these values to obtain a daily values, because you cannot really find daily values for KC, but the model can calculate the KC value per day from the values given for each growth stage. And also you'll be able to see that the evapotranspiration plot, that in the beginning of the season, the transpiration here is low. And as the plant grows, you can see that the red line start to be dominant, and the bare soil evaporation is less and less until harvest. And then the bare soil evaporation becomes dominant. You will be able to see also the irrigation with rainfall. And here, this is the amount of irrigation, but here you have rainfall added to the irrigation. The same for those events as well. When it comes to the crop yield, and this is really also yield obtained from the relative yield, and it shows this is the actual uptake, this is the maximum, and the ratio between the total actual to the total maximum gives you the relative yield. If you multiply it by maximum yield obtained in the area, you can get that line. But this is really for only scenarios, because we have already a proper calculations of the dry matter and yield here from the photosynthesis efficiency. And you can see here, this is the dry matter. And then if you multiply the dry matter at the day of harvest by what we call harvest index, you get the final yield at that point. The nitrogen uptake also, we calculate it from the soil N part. And you can see here that the water uptake maximum at the point really maximum vegetation takes place. And that's really gives you an idea how much the plant took from NH4, how much from NO3. And also the daily nitrogen leaching below the root zone. It gives you an idea. And also one important thing here is the balance. You have here a day water balance, and also you have here the salt balance, and you have the nitrogen balance. And most of the time, really, you need to look at these values. It should be close to zero. So to be sure that really all the calculations and input and output are really put correct. Presence of the drainage, we put the drainage here at certain depth, and we can look at the plots of the drainage. And at some stage also, you should be able to see the build-up of groundwater table. And also you can see the differences in the levels here as profile. Crop rotation, the model would allow crop rotations as well of different crops over the years, and you should be able to see that in the area where you have a crop, you will have transpiration. So there is a crop, here is a crop, in between the soil was left bare, and that's really the bare soil evaporation, what you see here as a blue line. So you should be able to run the model with crop rotation. And when you run the model with crop rotation, you should be able to see also the yield and the dry matter of every crop according to the rotation. The model performance indicators are is not part of the model, but most of the researchers, they use root mean square error or R square or CRM, and some others, they came with other parameters to describe the goodness of fit as well. So there are some non-conventional way to use the models. I came across from the users. Basically, my job really is not to use the model. My job is to develop the model, and a lot of people, they are using the model, and I discovered they use it with with something else in mind. I have seen people using it to measure to obtain difficult parameters like pi 50, which is the water uptake, uh, uh, the osmotic potential at which the water uptake is reduced to 50%. That's called pi 50. It's very difficult to measure. So sometimes they know the yield and they work out backward how much would be the pi 50 that would actually give me that yield that I measured. Some also people they get they work out the model to obtain the photosynthesis efficiency or even the leaf area index or the soil hydro properties. By knowing the yield or any other value measured, they work out what would be the value of that parameter they cannot measure or difficult to obtain from running the model. And also people, they run the model with either individually or collectively of parameters related to climate change, like CO2, radiation, rainfall, temperature, and to check the impact on the sowing and harvest dates, length of the season, the crop water requirements, and impact of seawater inundations and tsunami and the effect on the environment as well. I also saw some people that are using it to see the best crop rotation. For example, shallow rooted crops followed by deep rooted crops. And the idea here is to get the deep rooted crops to get more of the soil moisture and nitrogen that left below the shallow rooted crop for the season before. Also, some people they started to use mulching and organic matter, and they started also to use the model to look at the best irrigation scheduling and also to check on what's the best to use mixed waters versus cyclic fresh saline water. Also, I saw the model used to investigate the root system geometry, selecting suitable variety for crops for certain regions under climate change. And also I saw some use of the models to design a program for data collection. So these are few things that I came across based on the users, and I was really surprised to see that. So thank you. That's the first part of my presentation.
Thank you very much. But now it's second one.
Yeah. Now I will go to look at the model frames first, and then if we have time, I will just show some examples of applications by others, not by me.
Great.
Okay. So let me.
Participants are invited to register your questions to the professor, Dr. Ragab, and later all these questions will be addressed to Professor by Dr. Maria Kunieskova. So please formulate your questions in the correspondent window.
Is that appearing on the screen?
Yes.
Okay, thank you. So when I run the model for the first time, you will see that opening slide. So this is really what you will see first, and then it flicks to that frame. Okay. So that frame actually it's a combination of the field parameters and the global parameters. Global parameters gives you model run of the dates where your crop database of the soil and the crops, you can change that. And when you want the model to start from 2, and here the location of your results and the files that you will get in the data folder uh, related to. So you will have all these output. Um, and this is really the file outputs. And the fields here, you can specify how many fields, how many field or treatment you want to run, but you need to tick the box here to run. And here it will allow you to put your soils, and you can put these soils manually by using the edit. You can also have a new soil by clicking on new. And then you would, for example, if I click on new, then you will have to type here everything. And that will be saved. If you have soil and you look for equivalent soil, if you have a clay soil, but you don't have the parameters, you can go to clay soil and get a similar parameters, for example. And uh, you can also go for any type of soil, clay loam soil, and it will give you also where actually the soil comes from and the reference to all these parameters. It's the same for crops. Crops also, you can have a new edit, or you can look for crop parameters done by others. Here's, for example, faba bean from Syria, and it's there here from Raqqa, Syria, and you can also use this if it's similar to yours, or you can modify that. So that's really for the global parameters. Field parameters, for each field you specified here, you have to get your field parameters. So you will have here to specify if you have in the climate data, the radiation from climate data, or you have or you will use total and net radiation climate, or you will calculate the radiation from sunshine hours if you only have sunshine hours. And here you import the climate data, and you can clear, of course, the content here as well. Uh, and also evapotranspiration here, which I spent some time in the beginning about. So you have FAO modified, and that takes climate data to calculate the ET node. Or you can have the original Penman-Monteith, but here you will have to calculate the conductance by using the regression equation of Jarvis. Or if you have the APA, like Lisbon University researcher, then you can use the ABA, and you'll get the file of ABA and the leaf water potential. Or simply you assume, or in your region, there is one value that can represent the stomata conductance or resistance. You put it in there. Or simply, if you are measuring the conductance in the field, and we did actually in our work in UKCH in several years, we were measuring the conductance as well. So you can put this data here as well. For irrigation, the irrigation here, you can specify what sort of irrigation system. Is it basin irrigation? Is it basin with drainage at the bottom? That's we call it basin 2D, because at the bottom, the basin irrigation goes from 1D at the top to 2D at the bottom where you have the drains. You can also have border, center pivot TD if you have drains at the bottom. And you can have furrow, different type of furrows. You have rain fed, sprinkler, trickle line source, point source. And so you have also all those options for the different, and here you can also specify whether you have a drainage or not. And also PRD option here is also for you. And yeah, you can select, and then you import the irrigation file. Crops here, as I showed before, you have the common name, the maximum rooting depth, minimum rooting depth, the KC factor, KCB factor, fraction cover, the height, leaf area index. Pi 50 is osmotic potential at which water uptake is reduced to 50%. That's represent salt tolerance level. And here's also the crop can grow according to sowing date and harvest date, or you can, especially this is for climate change options. For people working climate change, the they prefer the crop to grow according to the cumulative heat units or degree days. So you can use that here, and you can run different scenarios. Here is the crop growth when we use the photosynthesis efficiency, and these are the values related to the equation I showed earlier. Those parameters, you have here default values. You can change them. Also, there is here water uptake effect, also stress factors, and this is temperature effect as well, minimum and the maximum for growth. Here is the rotation if you want single or you use crop rotation, and you put here the crop rotation one after the other and specify the year. Nitrogen here, it comes as the factors related to dry deposition, wet deposition, and plowing layer. And these are related to the uptake function of the nitrogen. And these are dissolution rates of specific mineralization, a specific rate of denitrification. All those parameters also are default, but of course you can change them if you have something measured. The general tab here gives you up to 4 layers of soils. So you can specify the thickness of each soil layer and the initial value of soil moisture content of the first day of the run, salinity initial of the first day of the run, the nitrate and the ammonia as well. So you need to specify those. And also here, the PF curve or unsaturated hydraulic conductivity curve can be given as input, or you can be given here as input. That's and then you have to select the file, or simply you calculate them from Van Newton equation, as I explained in the session before. So parameters here as well. These are parameters related to the area that you would like to see on the screen, the horizontal area, the vertical area, and the calculations of that. And it will give you really the values in the output files based on what you specify here as well. The profiles, you can specify how you would like to see your profiles on the screen. And also you can specify how you would like to see the values as output files. And you can also ask for range of the values rather than a point observation. It can give you an average value for a certain layer, and you have to select that. You can also avoid putting the results into Excel files by simply sending them to a database, Access database. Especially when you run climate change scenarios, it's advisable to use database because it takes more than the Excel files. Drainage here as well, you have also the distance from the irrigation, the depths below the soil surface, pipe parameters, and so on. And also concentration of salinity and ammonia and so on in the drainage water. So that's really, and then you go here and you run the model. So what I will do now, I will just get a file. It's a demo file. I managed to do it before noon today. And if now I go to global parameters, basically that file was eight fields. But for the time and to go quicker, I unticked those files. So I kept only one basin, one dimensional flow, trickle line source, and PRD fixed frequency, and basin drainage irrigation. So I put that here as 4 files only, or 4 fields. And now if I go here to, so that's really the field number 1, where the climate data is, the maximum, minimum temperature, sunshine hours, rainfall, humidity, radiation, total radiation net. And you have here the irrigation file. This is basin, one-dimensional flow, and the frequency is daily. This is, of course, not real data, this is hypothetical data. And also I put here if there is a fertigation. So here we have the rate, and the starting of the irrigation, the stop of the irrigation, so you can calculate the total amount. And here the fertilization, if you have fertilization happening, and also salinity of the water, and also nitrogen and urea if you have. Um, Then you can go to the crops, and here it's tomato, and these are the KC, KCB, and harvest, sowing date and harvest date. And also, if you want to put degrees, you can also put degrees here if you want to run the model with degree days. Crop growth, you can also have all these values for efficiency, and also you have to have here the harvest index for tomato, how much from the total biomass actually makes fruits, which is here 60%. Rotation is a single run for the moment. And also there is a nitrogen here, and nitrogen file can come here with all these manure and chemical fertilizers and so on. You can put, if you don't have, you can just put zeros. General, for simplicity, I only run it with one layer, which means homogeneous layer of 2 meters. Uh, and here we have a PF curve measured, so I can use it. Parameters here, also it's the dimension 1 by 2 meters deep. And profiles, this is what we would like to see on the screen, and this is how we are going to see in the output files. The drainage, there is no drainage in that one, but if you look at this one here, the drainage, there is values here for the range for that field. So that's really how the data looks like. And now if I now go to try to make a run, the first thing the model will do is to check on the data if it's complete or not, and then it will show you the screenshot where you can see the model run for a certain day, so the day can come here. And so that's really what's happening in basin. So now you can go and look for the other fields, what's happening there. So that's the trickle line source. There's the soil moisture plot as in real time. You can also go to PRD, and you can also have a look at what's happening here. Maybe that drip is the one that's operation for the moment. Now, at that point, the second or the left side of the drip line is now working, and that one stopped. And then if you go to the other field where you have drains, so that's really how it looks like. Then you can also go and have a look at different things like the salinity. You can look at the relative salinity, nitrogen. You can look at the soil moisture profiles, salinity profiles, nitrogen profiles, and you can copy to a clipboard. If you copy that, you can go to Word document and paste it there. So, for example, that if you really look at uh, so if you want to have a clip of this one, just simply you you look at a certain day that maybe you have a measurements for that day, as you are interested to see the evolution of the soil moisture or salinity, and then you can wait and then clip, and also you can pause the model, and you have here pause. But if now you clipped, you copy to clipboard, you can go to I'm trying to open a new Word document to paste it there, so you have it. So it is now pasted there. I don't know whether you will be able to see it or not, unless maybe I share it first. So there's really how it goes, and here you can have a look at the crop growth. Um, uh, Crop yield still early. Dry matter is still accumulating. The balance you can see on a daily basis, your balance, and it should be closer to zero, which is the case now. You can also look at nitrogen leaching, the nitrogen uptake, um, relative concentration, dry matter and yield, so moisture content, salinity. So you can really move around from one field to the other. So
Excuse me, Professor.
yeah,
We see the progress of the model running,
yeah.
but we do not see the Word file. This,
Okay, because the Word file needs to be shared as well.
or you can stop sharing this screen and maybe sharing the other one. We see that the model is working. Day 17 of 123. So
Yeah. What I was thinking to do before but I'm not sure if it's going to continue. But what I was thinking to do is to go to application examples while the model is running.
okay.
So that's what I was thinking to do now. See if I can do.
Excuse me. Maybe you can stop the model running, and then instead of sharing this Word Word document that had the results.
I'll see what I can do. Are you able to see that?
Yes, the PowerPoint. Yeah.
Okay, thank you. I left the model to carry on in the background. So anyway, this is just a few examples of really applications in different parts of the world. There are so many really, as you see, applications in different countries. I'm not going to waste your time on really going through all of them. And here really, so many countries, so many colleagues, so many friends and scientists did use the model and got excellent results. So this is example from irrigation with fresh saline wastewater applications. This is example from Napoli, Italy, where the saline and fresh water were mixed together on those basins here. And we were trying to grow quinoa and amaranth because they are saline tolerant crops. And it was a water coming from the well, and treatments were 25% of crop water requirements, 50%, 100%, and also saline water, the same 25%, 50 and 100. But the saline water was having salinity of 20 dCm per meter. So it has been really mixed and adjusted. The groundwater had been analyzed, and a certain amount of salts represents all the components of the groundwater. So we were not really adding sodium chloride to improve, to raise the salinity, but we looked at the concentration of different elements, and we added amounts from every element um, in the water to make that the water to be at 22.5. And that's my advice as well for anyone trying to adjust the salinity of water. Have a look at the composition of the water and don't add only sodium chloride, but there should be a balance between the sodium chloride and the other elements in the water. So we tried that, and we were able to test the model against the yield, dry matter, and the soil moisture and salinity, as you see here in the screen. And we actually were able to grow quinoa and amaranth in Napoli under these circumstances without really much reduction in the yield. So you can see here really we got a good yield under really 100% saline water. We got 3 ton per hectare, and 100% fresh water 3 and 3.1. So that's really quite indication of quinoa is able to grow in south of Italy and also is tolerant to that salinity. So that's one example of application. Another example in Denmark on weighing lysimeters in Aarhus University, where the salinity was ranging from 0, 10 decimals, 20 decimals, 30, and 40, and so on. And that was also quinoa. And we also got a good results really. It was a relative error percentage is really quite low, less than 3%. So that's also another good example. Another example for SALTMED, which is a greenhouse. The greenhouse issue also, I would like to draw your attention that the climatic data in SALTMED, it should be the one measured in the greenhouse. Some colleagues, they find a relationship between the climatic conditions in the greenhouse and outside, and they work out factors to use that. But you cannot really use the climatic conditions outside the greenhouse to represent the greenhouse. So that's one point really I would like to. But anyway, we used it here for pepper in Turkey, and we used also here, you can see here 1, 2.5, 3.5, 6 decimals per meter. And we managed to get reasonable yields, but it appeared to be sweet pepper was really sensitive, not very tolerant to high salinity. And also we followed the salinity or evolution under the greenhouse experiment. And we could also get the model to represent the field measurements was really a good matching factors. In Egypt, we also tried to use drainage water to irrigate tomato, and we applied drainage water mixed with canal fresh water, and also we applied the water as an alternating way. So we're comparing furrow and the drip, adding water in alternating way. So one time fresh water, the second time saline water, or we mix the two type of waters and we irrigate with mixed water. And also we applied that in Syria as well on tomatoes. And here in Egypt, we could see there is a slight improvement when you use alternate type of irrigation. So you irrigate fresh and then next time saline, slight improvement more than what you do if you just to mix the saline and irrigation water. And we were able to see the drip and the furrow actually predicted very well by the SALTMED. And this is Syria here as well. And it was also indicated that 7 decimals per meter would reduce the yield by 50% of the tomato that we used in both countries. This one in Italy here in Bologna, where we have wastewater. We got the wastewater from the wastewater plant here, and we have been spiking it with salt to bring it to 4 Siemens per meter, and that's really to try it with surface here, drip irrigation, and subsurface PRD irrigation. And we use the drip PRD. The pipelines were 12 centimeters below the soil surface, and we ran the model with this data for potato and for maize, and we looked at the salinity. And as you see here, the salinity and the soil moisture and the yield were nicely predicted by the model, and there were no much reduction really when it comes to the treated wastewater and 40 Siemens per meter salinity. Another also a good and new application that in Egypt, and that won actually the best paper award in ICID 2018 by Dr. Abdul Rauf from the National Research Center in Cairo, where he used the drainage water of fish ponds to irrigate the yield with different also nitrogen level, and to look at the possibility of using that water. Otherwise, it is wasted. And he got here that a good really yield when applying the drainage water of farms compared to when you use only fresh water. So there is a benefit really of using drainage water of fish farms on the wheat yield. Another also important thing that, and also unconventional way of applying SALTMED model came from also these papers from Egypt, from Dr. Abdul Rauf and his colleagues in National Research Center, where they were looking at the geometry of the roots and the volume of the roots, and they used the model SALTMED to obtain actually the root system and the root area, and they compared that, as you see here, with what they measured. In SALTMED model, basically, you can have that as part of the output. The model produces every day, actually, the widths and the lengths of the root system. What you see here as 0, it means this square occupied by roots. If you have 1, it means partially occupied by roots. If you have 2, it means the roots did not reach that yet. So this gives you an evolution. So the roots basically are here. That's half, of course, of the root system. The SALTMED calculates only half of the system or the flow domain and mirrors that to the other half. So basically you can do that and look at the geometry and compare it. And here it's comparison between grain yield and regulation.
Excuse me, Professor. There are many persons that have a huge interest in your model, and I thank you for your excellent presentation, and I will pass the control to Dr. Maria Kuniskova, our coordinator at FAO, for making such questions to you, if you are so kind. So, Dr. Kuniskova, the floor is yours.
Thank you, colleagues. So there are many questions that were raised in the Q&A chat box.
Okay.
So let's, I think that we will need around 15 minutes maybe to address all of them. So we ask our excuses from the participants if we take a little bit more time. So the first question was, what is the main objective of the model? So I understand that the title of our webinar is more or less the answer to this question, but I would like you maybe to give some sort of your understanding what is the main, let's say, goal of this. What is the main parameter that you model with this model?
Well, it is really, it looks like a simple question, but it is the whole model. And I left to the first slide about why you use SALTMED. What's the benefit of using SALTMED? It is a tool for management of crops, water, salinity, and it is not only for field practitioners, but also for academic researchers, especially those who are working, for example, for hormone biochemical signals like ABA and so on. So there are people, they are working on research, and you will find something for them in the model to use it for. There are some people who are interested in the yield only, and those mostly the practitioners. And those they can also just simply put climate data, irrigation data, and they can have a yield, and they can run the model with what if scenario. Instead of doing so many treatments in the field, they can calibrate and validate with 1 or 2 years, and then they can, after that, once the model is calibrated and got the right soil and crop parameters, they can run it with what if scenario of different management, different nitrogen levels, and so on. I can give you example of a paper from Pakistan. They found that the climate change is published anyway. I was about to talk about it, and that paper basically predicted a reduction in the yield by 2090. But at the same time, they said that reduction can be counteracted by adding more nitrogen. And that's the whole thing is scenarios. So they didn't add that amount of nitrogen, but they are trying to see if there is in the future reduction in the yield, how can actually avoid that reduction. And they came with this idea, if I run the model with the climate change and more nitrogen, would I be able not to lose any yield? And that was published actually recently. So there are ways that people, they use the model also to predict as well without doing field experiments.
Okay, thank you. So some participants report that they face issues while installing and running the model.
Yeah.
So the question is there any ongoing work to improve the stability of the work of the model? So, and is there any work ongoing?
The model comes with a standard way of installation. And what actually the users do, instead of allowing the default folders where the data should go to, they try to install the model on G drive or F drive or somewhere else, where actually the model is designed to talk to the database and to talk to some other supporting software, like those software they make the diagrams and the pictures, and those usually installed other program files, and usually program files normally under the C drive. So my advice to avoid this, allow the model to install the folders as described while you are installing. It will tell you where everything will go. Just accept what the model says, but not over rule the model default destination.
So in this regard, is there any contact or like a address where people can
Yeah, we have
contact?
I mentioned in the YouTube, there are how do you call that, troubleshooting problems and solutions, and that actually was done by a professional company. It's called the STEP in Germany, and these are actually most of the issues that people reported are in there, and they should look at. There are about 10 issues I came across, and I put the solutions for them on the YouTube, so people they can see visually and visibly as well how the problem is solved using the YouTube. If anything else, they can contact me by email.
Mm-hmm. Yeah, so the same style of the questions is if it can be run with Python, And also about the Windows, as soon as it is now for Windows 7, 8, and 10, is there any upgrade of the model available?
The model for the moment runs with Windows 7 and above until 11. We don't know what Bill Gates is going to do with the new Microsoft. If he comes with another Windows and so on, then I will have to go back to the software developer and to make it compatible with the new Windows. But for the moment, the model runs with all the Windows above Windows 7. It doesn't run with the Windows like Windows 95 and that sort of thing.
Okay, older versions, but also
Yeah.
works. And Python?
No, we have nothing to do really with Python because the model actually is part of it is written in C++ language, the mathematical part, and the interface part is Visual Basic.
So next question is related to the practical use of this model for the farmers. Is there any option how to simplify the model so that farmers can also use it? Any option to include soil moisture sensors, for instance? What do you think?
Well, the model can accommodate soil moisture if you have soil moisture sensors, and we had that in the experiments actually of those projects. We have been measuring everything basically. Yeah, it would allow that. Uh, of course, you have in the model you have options, and you have seen so many options for any uh, uh, academic researcher will go for the full, of course, capacity of the model. But if really some younger professionals or someone from extension services, they will have to use part of the options. For example, the model has got salinity and nitrogen. You don't need to run the model with salinity and nitrogen. There is two little windows there. You tick them, and it says skip nitrogen, skip salinity, and that will only make the run to run only with water. You won't waste your time and your memory, and the model will go a lot quicker. So you can run the model without nitrogen, without salinity if you don't have them, and also you can use simple options. You can simplify. The model has so many options. If you want to go for the Rolls Royce, uh, you can go. If you are happy with the Fiat, you can go. So the model has so many options to offer.
Okay, thank you. And what is the required data to build a model? So you just said that you can skip many, but maybe there is some minimum set of data that is needed for the model.
Well, the minimum really thing is that climate data, because the driving really force behind the model is the climate. So you have to have this climate data, and of course the water. The rest can come from database of the model. The model has got two databases, one for soil and one for the crops. And really, the most important things is really these two files, is the climate data and the irrigation data.
Thank you. So next questions, they are more specific, more technical, let's say, related to the model. So one of them is that, does this model, SALTMED model, help to calculate how much water is needed for rice field during the growing season? Can you use this model to calculate?
Yeah, yes, you can. It's one of the strengths of the model that you can calculate the crop water requirements, and that actually it comes as a separate column in the output file as crop water requirements, and you can look at that. Yeah.
So you mean that you input the rice, the crop, and then it can calculate?
Yeah, you can run the model with the data of climate irrigation, and you can really see how much the crop water requirements. And you can also use an old data from year before or something, and that will give you also an idea what to expect for certain months as amount, and that's helpful to the reservoir managers. So if I tell the reservoir manager I'm expecting the rainfall of this year to be similar to last year or 10% more, the crop water requirements of last year was that I will need 10% more because the rain will be less or whatever. So you can actually get an idea as how much water you require from the reservoir.
Thank you. So next question, I suppose that it was asked before. Actually, you presented this part of your presentation about the root growth model. So is this used, the root growth model, as it affects the salinity concentration in simulations and in interaction with soil texture? So the answer, I suppose, is yes, but maybe you can elaborate more what type of the root growth model is used.
There are some parameters related to the root growth in the model coming from the database, which represents the maximum and the minimum rooting depths. So that's one thing. The other thing is that there is also a parameter about the root width to the root depth ratio, and that's input from the user. And that depends on actually the soil texture. Usually, if you have really a clay soil, you will have, of course, the root will not penetrate that deep and also might not be as wide. So there are three parameters to describe the root system according to the soil conditions. And we take that and we interpolate it on a daily basis, like the KC factor. You get the KC factor per growth stage, and the model will interpolate between two stages to get a daily values, because the model simply runs on a daily value. It doesn't run with stage value.
So thank you. Next question is very specific, but I will read it as the participants are interested. So I have used the model to simulate raised beds, but I couldn't simulate the width and the depth of them. And when simulated the drip irrigation system, I couldn't change the distance between the emitters and the lines of the dripper with each dripper. So maybe you can advise how to
in the irrigation file,
simulate this.
in the irrigation file, there is something about the depth of the irrigation system. In the irrigation, not the file, the irrigation dialog. When you open the model, you should be able to see where you put actually the model is still running, but anyway, I think there is a depth for the drippers, and also there is maximum depth for evaporation from the soil surface. And also there is something else related to the depth of accumulation of water on the soil surface to allow some excess runoff. So I don't see why you get stuck at that.
Yes, so maybe it's because of the raised beds. So I'm not sure, but maybe it's because they used in a different type.
It can be the units. It could well be the units. Maybe the units is crucial here, because for example, for the furrow irrigation, it is a liter per hour per 1 meter of the furrow length. Some people, they forget about that meter of the furrow length. So it's a liter per hour per meter length of the furrow. And the same for line trickle source as well. Maybe the units is not correct, but I don't think really that's reported by anyone else really.
Okay, so next question is about N2O. So I'm conducting long-term N2O experiments with a focus on crop rotations. And so can SALTMED model help with analyzing N2O emissions and crop rotations effects?
Yeah, I think I showed that in the slide where you can run the model with the crop rotation, and even cleverly you could run the model with shallow rooting depths followed by deep rooting depths, and you look at how much actually nitrogen was taken from the deeper layers by the deep-rooted crop and so on. So yeah, there's no problem at all. And I showed the slide and the screenshot from the rotation.
Yes, thank you. What is the flow area in irrigation tab, and how to calculate this parameter? Flow area.
How small?
Flow area. The participant is asking what is it and how to calculate this parameter.
Not clear about.
Irrigation tab, there is a parameter which is called flow area. But we can keep some questions and address them after I will send after the webinar.
No, I didn't get the question. Can you
Yeah, because
say it
they're
again?
very
Or can you come
specific.
closer to the microphone, maybe?
There is a parameter which is called flow area. Flow area.
Yeah.
It's present in irrigation tab.
Yeah.
So, and the participant is asking how to calculate this parameter.
The flow area is actually explained in the guide, in the model guide, the model user. What actually it shows the area you will see on the screen, and that's really explained how the model works. What you see really is half. I mentioned that before. We calculate half of the flow domain, and we mirror that, and that's actually saves half of the memory, and also makes the run to go quicker. So we assume the left and the right half are the same. So we run the model numerically for one half, and we mirror that to the other half. And we try to work on 1 meter square when it comes to basin irrigation, for example. That's what you see sometimes. So if you see a plot, you will see the middle, and maybe I can show you that if I can. The model is still running, but I think I should, model run successful, okay. Do you see that screen?
Yes, like this. A lot of info. So
Okay.
I'm
so
sure that
what you see here in the screen, for example, you see that point here, it's zero.
Stop sharing. It was visible on the screen, but now we stop sharing.
Okay, sorry. Do it again. Do you see that plot here in the screen? You have here zero,
yes,
and then you have 1 meter here, right?
yes.
And then that's also mirrored on the other side as well. So basically, yeah, that's the flow domain is you have also 1 meter deep. So 1 meter deep that direction, and 2 meters deep here, and 1 meter here. But we calculate only a half of that. That's the flow domain.
Well, thank you very much, Professor Dr. Raghav, for your excellent presentation, and congratulations for your model. That is a comprehensive model that takes into account climate, irrigation, plant, soil, and as well, that is very important, management options, as for irrigation and fertilizers and so on. And as you demonstrated, the applications of the model could be very varied, and I'm sure that the participants will address you some other questions that we can convey to you for advising them, and some of them are very interested in contacting you for specific questions or proposing you maybe some kind of collaboration or activity as advisor.
Yeah. Here
Thank-
the model is finished, and maybe it's now time I can show the remaining participants how actually the final yield looks like. Crop growth. This is really the KC and KCB, and this is the final after the end of, and this is the nitrogen uptake final, and also you can have here the crop growth, salinity indicators, the dry matter, and here you have the final yield here, for example. So this is really the final. This is the irrigation of the season. Have the evaporation here also of the season. So this is now really the final run of the 4 fields. Just I wanted to show the participant how the model at full run.
I have a suggestion that maybe you can copy this screen in a Word file, and
Yeah.
then we will distribute this to the participants, and so they have time to see in detail the results and maybe address questions to you. So thank you very much. It has been a very outstanding webinar. I was watching the comments on the question and answer, and as well in the chat window. People is really interested in your model, and I'm sure that this model that is well used will be even more used by new researchers, academics, and colleagues. So again, thank you very much, Professor Dr. Raghav, for your collaboration with this network. We appreciate that you are honorary president of another network that is related to us because irrigation is an important part of the activity in the salt-affected areas and the risk of salinization. And we expect that the collaboration between these 2 networks could go further and for mutual benefit. Thank you very much, and I appreciate very much your effort physically being today with us, even affected by this inconvenience. Thank you very much.
Yeah, thank you for inviting me.
Thank you so much.
It was a pleasure.
Thank you. Maria, could you please put the questions and also
Yes.
the chat box contents to me so I can thank people as well?
Yes.
That would be great if you can.
So,
anything else, of any email message or whatever related to questions I didn't answer, I can also answer by email.
So thank you very much, Professor Raghav. Thank you very much, Dr. Maria Koniskova, and to all colleagues that were with us today. And we hope that this webinar could help for capacity building and for the progress of your research. Thank you very much. Keep in touch. Those that are registered to the network will be regularly updated of the webinars and other activities that will proceed. Thank you, and have a nice rest of the day.
Thank you.
Bye-bye.
Have a good evening. Bye-bye. Bye-bye.